Hey guys, so this is the last video for chapter 8. So far we've learned about Mendel, we've learned about dominant and recessive traits, we've learned how to make a Punnett square and how to interpret the results of a Punnett square. And now we're going to talk about excuse me, the exceptions to those laws. We talk about the law of segregation, sorry. We talked about the law of independent assortment, but we said that there's going to be exceptions to those laws. There's going to be exceptions to the theory of discontinuous variation that Mendel came up with. There are exceptions every time we make rules. So one of our first exceptions is incomplete dominance. Incomplete dominance is where you are going to have both alleles kind of show up. We're going to have them both kind of show up, but half haphazardly. Just if you had a red flower and you had a white flower, or purple, right? If we had purple like Mendel had and white flower, then you are going to end up with an incomplete dominance or continuous variation like they thought back in the day, a pink. Oh, wait. Oh my gosh, sorry. So with incomplete dominance, red kind of halfway expresses and white kind of halfway expresses, and you end up with this pink version of these genes. So this is incomplete dominance. It's not red, it's not dominant as red, and it's not dominant as white. It's incomplete dominant, and you get half red and half white, you get pink. So I'm going to need you to know these exceptions. I'm going to need you to know examples of the exceptions. So a red parent flower and a white parent flower producing a pink offspring in the F1 generation would be an example of an incomplete dominance. Blood types. Do you know your blood type? I know my blood type, and you too can know your blood type if you are able to donate blood, or if you have ever received blood. They will type you before you receive blood to make sure that you get the right type. How do you have the blood type that you have? Well, I knew because I went and donated blood at the Red Cross blood drive that we just had last week, by the way, at Georgia Highlands campus. And they also are going to test for antibodies to COVID, which is pretty cool. So I'm going to see if I have those or not. But how do you know what blood type you have? Well, there are a few different blood types that are out there. There are some rare exceptions, but these are our most common blood types. Blood types are determined by two words that both start with A. There are antigens and there are antibodies. Antigens are these things. They are located on the cell membrane. Remember we talked about coatings on the outside, like the glycocalyx, that was the sugary coating on the outside, and it gets attached to proteins. It could be attached to the lipids, the phospholipids. And so we are going to have things. Oh my gosh, what's wrong with me? We are going to have things extending from that cell membrane, and these are antigens. And so if you are an A blood type, you are going to have A antigens on the outside of your red blood cells. And you, if you are A blood type, you do not want B. You don't want B blood type cells. You want B cells because you are A cells. So you're going to create antibodies. Here's your antibody against B. And here are your A antigens. So if you are A blood type, you can obviously receive A blood, and you can also receive O blood. We're going to talk about why you can receive O blood, and it is because it doesn't have any antigens. There's O, O's got nothing. Zero, nada, nothing. No antigens over here for O. B is the exact same thing as A, except you have B antigens instead of A. So these are all A antigens, A. These are all B, B, B. And if you're a B blood type, you don't want A, so you're going to have antibodies against A blood type floating around in your plasma. Also, they'll call the very technical term liquidy part of your blood. Your blood is made up of formed elements, red blood cells, white blood cells, and platelets, the big, thick, chunky things. And then you're going to have liquid parts in your blood, and that's plasma, and that's where your antibodies hang out. So if you were B blood type, you are going to have antibodies against A in there. You can also be AB, and that is an example of codominance. Codominance. Before we talk about incomplete dominant, where something was kind of halfway showing up. With codominance, both display. You have A antigens, you have B antigens, and they both display. So this is codominance. Here we have A's, and here's B, and here's an A, and here's a B. You have AB blood type. If you are AB blood type, you don't want antibodies against A because they'll latch onto your blood cells, cause agglutination, and cause a clotting disorder. So you don't want to create A antibodies because you have A antigens. You don't want to create B antibodies because you have B antigens. So there are no A or B antibodies on a person that has AB blood type. So AB blood type is called the universal recipient because it can receive not only A blood, it can receive B blood, it can receive AB blood, and everybody can receive O blood, which is pretty cool. Um, this last blood type is O. O, you see, there's no antigens. Like you got these little sticks coming out here, but there's no A, there's no B, there's nothing there. So if there's nothing there, it is recessive. It is recessive. O blood type is recessive. It will defer to red. It will defer to A or B if given those genes. If in a heterozygous situation, it's going to be recessive and it's not going to show up, but it has to be homozygous and you will see these show up. So this is O, it does not have antigens. Therefore, antibodies are present, antibodies against A and antibodies against B. O is going to be considered your universal donor. With blood types, you're going to have positives and negatives. So that's an RH factor. You may also see it called factor D. RH for rhesus, for the rhesus monkey. And if you are RH positive, you would be A. And then you would have other antigens for the RH. Antigens for RH, this would be A positive. What if you were negative? If you're negative, then you're not going to have the RH antigens. So I'm not going to draw RH over here. There's no RHs here. So this would be B negative. So technically, our O neg is our universal donor. And our AB positive, does have a positive there, is going to be our universal recipient. Since O is our universal donor, these guys are lacking, our O negative is lacking the rhesus antigens, and it's lacking the A or the B or the AB. That neither one of them show up at all. So those are the different blood types. We said exceptions to the rules was codominance, and that codominance is this blood type right here and right here. It's AB, excuse me, blood type. So you may need to be asked to give a Punnett square, know how to interpret that Punnett square, or also understand that AB is codominant and it is an example of codominance. What if we did a Punnett square with different blood types? What if mom is O? Mom is me. I am mom. So I have O and O is recessive. What if dad is a different blood type than mom? What if dad is B and he's heterozygous? So if dad is B, we're going to write his dominant B because B is dominant. A is dominant, AB is codominant. If dad is B, but he's a carrier, remember carriers are heterozygous. So we put a little recessive O over here. If dad wasn't a carrier, if he was homozygous dominant, we'd put both B's, but we're going to say dad's a carrier. Dad is a carrier, B and O. Let's write dad's stuff, B, B, O, O. We'll bring down mom stuff, O, 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 O. What is the likelihood of these two parents having an offspring with type O blood? Well, both of these, we got two, and we got two. So it is a 50% chance that these two parents will have a type O blood type. It is also a 50% chance that these two parents will produce a child with type B blood, just like dad. So it's a 50-50 shot with these parents. What if dad was A, and we're going to say he is, let's make dad codominant. Let's make dad AB. All right, so dad's codominant, dad's AB, and mom is still O. I am O. So I'm going to write O there, and we're going to say dad has AB blood type. So dad has A. We got to put the B in the B. And then what is the likelihood of this parent combination producing a child with an O blood type? 0. There's 0% chance with these parents having a type O blood type. O, 0%. What about AB blood type? There's none on here that have both A and B, just like dad. So it is 0%. What about A blood type? 2 of them, these two, this one and this one, are A blood type. So we have a 50% chance with these parents having an A blood type. And same with the B. It is 2 of the 4, or a 50% chance of these parents producing a child with B blood type. But neither parent will have offspring that are just like them. So interesting combination with blood types. But remember, O is recessive whenever you have to set this up, if you have to set this up on the test. And remember, A is dominant, B is dominant, A and B both expressing is co-dominant. Moving on. Yes, blood type has multiple alleles. You could be A, you could be B, you could be O, you could be AB. So definitely multiple alleles. Just like over here with coat color with these little bitty rabbits. You could be brown, you could be black tipped, you could be white with the black paws with a cute little pattern on it. You could be completely missing coloration, and you could be albino. So lots of different alleles with coat color, and rabbits with chinchilla and Himalayan and albino. Other strange exceptions, sex linked. So we mentioned in previous videos that there is a karyotype, and we have, as humans, we are going to have 23 pairs. Oh my gosh. Clearly I need some caffeine. We are going to have 23 chromosomes, and out of the 23 pairs of chromosomes, 22 of them are autosomal and 1 was sex chromosomes, right? And for humans, we have X and Y chromosomes for sex chromosomes. If something is located on the X chromosome or on the Y chromosome, it is considered sex linked. Sex linked. So we have Khan Academy link to check out. For more on these sex linked, you might be asked to perform a sex linked Punnett square on a test. Here we have some set up with fruit flies, and it has to do with their eye coloration. Their eye coloration genes are located on their sex chromosomes. So you can tell based on their eye color how that's going to play out. Let's talk about another one. Here we have, all right, a sex-linked one where dad has the dominant allele and mom has one dominant and one recessive for the X-linked disorder. So we're going to say that we will say that big C equals normal vision and little c is going to equal colorblind, because colorblindness is located, red green colorblindness is located on the X chromosome. So in this case, we have mom and dad. Let's play it out. Mom, I always put mom on the top, but again, you can put mom on the side. Mom is a carrier with her 2 X's. Again, remember, always write the chromosomes first. If I'm putting mom on top, mom has 2 X's. If we're putting dad up here, dad only has 1 X. So don't forget, this is one of the most important parts of setting up the Punnett square for a sex-linked disorder. What are the sex chromosomes? Mom, 2 X's, dad, an X and a Y. And when we talk about these sex-linked ones, all we're focusing on is the X. That's the one we're focusing on this day, because colorblindness is located on the X chromosome. So In this situation, we have mom up top, we have dad on the side, and we have mom is a carrier for color blindness. Mom is not colorblind because normal vision is dominant, but she's a carrier. So here's mom's big C and little c. And dad is going to have normal vision. So dad will have a big C. And now we're going to bring these genes down and across just like we normally do. So I'm going to write mom's first. Mom has her X's and a big C and a big C. Mom has this X and a little c and a little c. Dad is going to only have one X. He only has one X to contribute, and that's all he's got. All right, his Y's and his X. Dad's X is a big C. This X, big C. What is the likelihood of these parents producing a colorblind male child? A colorblind male child. So all I'm looking at, if we're asking about the likelihood of producing a colorblind male child, all I'm looking at is the males. So I'm down to 2 boxes out of the 4. These 2 boxes are the males. What is the likelihood of producing a colorblind male? Well, one of these has a big X, a big C on its X. So that is normal vision. That's not going to happen. So this one is normal. And this one over here on its X is going to have a little c. Remember, this little c is colorblind. So the likelihood of these parents with this genotype, with mom having heterozygous for colorblindness and dad's only got the one gene, but he has normal vision, we are going to end up with a 50% chance that they will produce a colorblind male, a colorblind male. So on the test, if you have to do a Punnett square for sex linked, write your Punnett square down. Write your mom's X's. Remember, mom's got 2. Write down dad's X and a Y. Then find out what genes go on those. Is mom a carrier? Is mom homozygous recessive? Is mom homozygous dominant? And put her genes on there. What is dad genetic for? What genes is dad bringing to the table? Is his X chromosome going to be dominant or recessive? Remember, he's only got one. So what does dad's genes look like? What does mom's genes look like? And that's the biggest, to me, the biggest hurdle out of setting this up. Then it's just interpreting the results. If we were asking about offspring in general, we're going to look at all 4 boxes. If we are asking about just male offspring, then we're only looking at these 2. Just this one row here are the males. All of these are the females. So if asked about the likelihood of passing on whatever it is, hemophilia, colorblindness, whatever, muscular dystrophy to the male children, then look at just those lower 2 boxes in this case. Just look for the male 2 boxes, and then what's it look like in those 2 boxes? Is everybody got it? Is it a 50-50 shot? Does nobody have this thing that we're looking for? Know how to set this up. To me, this is the trickiest of the Punnett squares that I'm going to ask you to do on the test. So understand how to set up the Punnett square. Understand how to set up the sex-linked Punnett square. It's the trickiest. And once you've set it up and you run it through, know how to interpret the results. Remember, only 2 of the 4 boxes are males. Only 2 of the 4 boxes are females. What am I asking for? Am I just looking for males, or am I looking for just females? I'm looking for all the offspring, and I'm looking at all 4 boxes. Is it a 50-50 split? Is it a 25 out of, is it a 1 out of 4 chance if I'm looking at all the offspring? Am I looking at just males and it's 100% of the chance? How do I interpret those results? Please practice these, play around with them, do them at your own home in your own notes, and let me know what you're getting and if you're running into problems, because you will see these on the test. Here are examples of different things that are found on the X chromosome that are sex-linked. Colorblindness. If you are red-green colorblind, all of these dots look the same to you. You cannot make out the number found with the different colors in this picture. Hemophilia is a disorder. It is genetic, and it is when you are lacking certain clotting factors that help you clot. And you run the risk, if you are hemophiliac, of bleeding out due to internal bleeding, or if you had an external wound. Either one, you cannot clot and keep your blood in hemostasis where it needs to be. Hemophilia was very prominent in royal families in Britain and Russia, all sorts of royal families, because you were going to keep the bloodline pure. You weren't going to breed with outside family members. So recessive traits really showed up, and hemophilia was seen a lot in royal families. And then muscular dystrophy is also another sex-linked disorder found on the X chromosome that is going to lead to muscle weakness. It's degenerative in life. Other exceptions. Some of these things we've been looking at, and it's just one gene. One gene, it is a yellow pea or it is a green pea. It's wrinkled or it's round. It is just simple. That one trait, that one allele is coded for by that one gene. But of course, life is not simple and things are complicated. So one of the traits that is complicated is skin color. Skin color is polygenic. Poly means lots, or many. Poly means many. So polygenic, your skin color is an example of polygenic inheritance. Lots of genes are going to code for your skin color on your person, on your body, what shows up. So of course I have another family picture to display this. So this is my family. This is my Uncle Jerry, and this is my Aunt Elaine, and this is their child, my cousin Karen, and then Karen married BJ, her husband. Carrie married BJ, and they produced their child, BJ Jr. So here is BJ Jr., BJ Jr. over here. So if we are looking at skin color, we can see that my aunt and uncle are about as white can be. And they produce Karen, who looks to have a remarkable tan in this picture right now. I don't know why she's so tan in the picture, but Karen is pretty white. She's like me. And you can see that BJ is on the darker spectrum. So when we have a parent that has one skin tone and we have another parent that has a different skin color, then we are going to have that again controlled by lots of different genes. What is the likelihood that their offspring are going to have genes just like mom or genes just like dad? We said that because of the law of assortment, independent assortment, and um, we everything has an equal chance of expressing. We can run the math on this. When we run the math on this with mom with this skin coloration and dad with this skin coloration, and we run that math, we are going to see that there is a chance that the offspring could look like mom or could look like dad, but the chance is rare. It's more likely that their child, like BJ does, has this beautiful caramel skin color right in between there. It's more likely. So when we crunch the numbers about skin color, which is controlled by multiple genes, we will see that the likelihood is somewhere in the middle. It is more likely to have somewhere in the middle, but it is possible. It is possible to produce offspring that will look just like one parent or the other. It just depends, but more than likely, the offspring will have some sort of middle of the road in this trait of skin color, which is an example of polygenic inheritance. Um, other things that we need to talk about. Linkage. So we said when we learned about meiosis, that meiosis was super cool, that we're only going to um, talk about meiosis when we're making gametes. We only use it for sperm and eggs. And when we undergo meiosis, there was a really critical stage. And it was, this my cat is showing up here. This is Smudgies. This is Smudge. Oh, he's so handsome. Prophase 1 was where crossover happened. It was critical because if we didn't have crossover, we didn't have genetic variation, and those gametes would all look just identical. They would all look like mitosis happening. And we need crossover. We need genetic variation. It's the whole benefit of sexual reproduction is to get genetic variation. So crossover was happening at prophase 1. It was super important. But when we discussed um, those laws, when we discussed the law of segregation, that everything separates equally, and the law of independent assortment, that some genes don't influence others, when we mentioned those laws where everything separates equally and everything has an equal chance of passing on, linkage is a little bit of that exception to the rule. And what happens is when we undergo crossover, where you're located on the chromosome kind of matters. If you are located really close to another one, it is more likely that you will be linked in that chromosome and that those two genes will be passed together as one unit. So in this example, we have a beautiful picture of crossover, and it might be possible that both B and A crossed over together as a unit. And if that happened, we would consider those linked. That if they crossed over together because they were really close together, and this one um, pulled off and swapped over with these two, then we would say that those genes are linked. There's a more likely chance that B and C would be together. A is much further away on the chromosome than C, so it's less likely that A and C are linked. You probably won't see those guys together, but there's a chance that B and C may swap out together. And if they do, then that's considered linkage. Um, Epistasis is another exception to the rule. We said in the last law, the law of independent assortment, that every gene is separate, it is independent, and it doesn't affect other genes. But in epistasis, some of these genes are going to have antagonistic relationships to the other. In this case, we have mice. We have mice and we have coat colors of the mice. So there's one gene and it is gene C. And gene C is going to be, oh, excuse me, if you have the dominant capital letter C, you're going to code for coat color. You're going to actually express whatever genes you have for that coat color. So big C is going to be, you will have a coat color. Little c is no coat color. And then we have A is going to be the actual color. And there are dominant colors of agouti. There are recessive colors of darker black. And then there's going to be albino where you don't have that. So this guy has a capital C. Coat colors expressed, 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 expressed. We have capital C's in all of these. We have at least one dominant C. So coat color will be expressed, and that coat color will then either be, if it's a dominant A, then we're going to have the agouti color. If it's a recessive A, then we're going to have the black color. But what if we don't get the dominant C? What if we get the lowercase c, the recessive C? If we get the recessive C, remember that codes for coat color. So it doesn't matter if you have agouti coloration or if you have black coloration, you're not going to have any coloration because you have recessive Cs. That is an example of epistasis, where one gene is going to block the expression of the other gene. So if C acts up, then A doesn't even happen in this sense, and that is epistasis. So please let me know if you have questions about this chapter. It is, to me, I feel it's complicated. I also feel like it's really fascinating, and I could play around with this stuff all day long. I could talk about this stuff for way longer than I really should have in this video, because we've probably done, I don't know, like an hour and a half, 2 hours worth of video on this chapter. So I find this really fascinating. If you have questions about this, please practice Punnett squares on your own and let me know if you have questions about these things. It's really cool, and I want you to feel comfortable with this. So practice, practice, practice. Do your study guides and let me know if you have questions from chapter 3.